Hello! Welcome to Biological Psychology. In this video, we're going to explore biological psychology at a micro level with a special emphasis on neural communication. We'll begin with a familiar topic, one that we introduced in another video. This is the concept of the synapse, the junction across which a nerve impulse passes from an axon terminal to a neuron, muscle cell, or gland. As usual, a picture will say 1,000 words. In this picture, we have the schematic of an upper neuron, which we will refer to as the presynaptic neuron. And also here in yellow again will be a postsynaptic neuron. As you might remember, this being the bottom of the presynaptic neuron, we're getting a look at its axon, and this being the top of the postsynaptic neuron, we're getting a look at its dendrite. And in between, we have a small unit of space, this junction, that we refer to as the synapse. Also, you might notice that we have little packets here of neurotransmitters that are going to be leaving the presynaptic neuron, coming into the synapse, and being received on the postsynaptic neuron's dendrites. There's an interesting analogy between the operation of these types of neurons and a simple battery. In fact, we'll develop this metaphor by thinking of neurons as if they are tiny batteries. To appreciate this point, let's just remind ourselves that here we have a AA battery, and the AA battery has a small knob on top for the positive charge, and down here on the bottom there's a negative charge, or a unit for the negative charge. And we have in the center here an indication that this is a 1.5 volt battery, and for scale we have a simple match, and that match might be something on the order of 3 or 4 centimeters long. We have very similar principles occurring inside of a neuron, where we're going to have a positive component, that will be the outside of the cell, and a negative component, that will be the inside of the cell, and this will be happening over a much smaller range of space, not 3 or 4 centimeters, but something on the order of maybe microns or millionths of a meter. Likewise, because these are tiny batteries, we're not going to have a 1.5 voltage difference between the top and the bottom, or between the positive and the negative, but rather than 1.5 volts, we might have just a small number of millivolts, or thousandth of a volt. Remember, these are tiny batteries, but there are similar principles between batteries and neurons. Let's learn a few more terms before we take a look at a schematic of a neuron. One important term is the so-called resting potential. This is the membrane potential of an inactive cell. And when we speak of potentials, we're talking about the relative preponderance of positive ions versus negative ions, a simple difference score. The electrical state for a resting potential, typically in neurons, is around minus 70 millivolts, as an approximation. And specifically, the inside of the neuron is negatively charged relative to the outside. Just as we saw back here, we might typically have a positive charge up here and a negative charge here. For the neuron, we have a difference between the inside and the outside, and that difference is approximately 70 millivolts. We have some synonyms when we're talking about resting potentials, and these include such phrases as the resting state, or the stable state, or the polarized state. You might hear neuroscientists and psychologists talk about these 3 terms interchangeably, always being used synonymously with resting potential. Okay, so let's zoom in and see if we can further develop our battery metaphor. So, here what we have is a cell membrane that is separating the negative side, again shown on the bottom in this orange-pink type color. You might see small negative signs here as I'm moving my cursor. And on the other side of the cell membrane, we have positive values, and these are in the extracellular fluid. That's the fluid outside of the cell. Here's the fluid inside of the cell. We also call this cytosol. We can begin to make some measurements to determine the resting potential. You might notice that we have in our apparatus a reference electrode that is floating in the extracellular fluid and picking up these relatively positive charges. And then we have on the other side an electrode that's being inserted through the membrane and picking up mostly these negative charges. So if we measure the inside relative to the outside, we would have a difference, a potential, that would be at around minus 70 millivolts in this schematic. So not too different from the principles that we had seen in the battery in the earlier slide. Another important kind of potential is called the action potential, and we contrast this with the resting potential that we saw in the previous slide. The action potential can be defined as a short-term change in the electrical potential that travels along a cell, such as a nerve or muscle fiber. A common threshold to launch or begin an action potential is approximately minus 55 millivolts, and this might vary slightly from animal to animal, but for many mammals, that range would be between maybe 55 millivolts or 60 millivolts. We have some synonyms also for the action potential. These are called nerve impulses. We refer to this also as firing of the neuron, nerve firing rate. We call it spiking, a spike or a spike rate. We also refer to it as a depolarization in contrast to the polarization that we had for the resting potential. Let's now take a look at how we might graph an action potential. This is slightly busy, so why don't we unpack it one bit at a time. Let's start on the y-axis, where we are tracking the voltage, again expressed in millivolts, but it's the same principle as we had seen in our battery. Here's the 0 mark, so this would be perfectly neutral. Here we have positive 40 millivolts. Here we have some negative values, and some important numbers for us are going to be minus 70 and minus 55. We'll come back to that, but that's the millivolt axis. It's tracking our voltage. On the x-axis, we're looking at time, and time is now in, again, very small units. It happens to be milliseconds. So we'll be looking at the change in voltages across millisecond time scales. Okay, so as we said in the preceding slide, a resting potential is typically around 70 millivolts, that's negative 70 millivolts, and we can have some fluctuations that are moving around that general number. And every once in a while, a stimulus will be presented to the animal, and that animal might have a response to the stimulus, which is signified by a slight increase in the millivolt value, but not quite up to the threshold of minus 55 millivolts. Here comes another stimulus, perhaps, and we get yet another increase. We get a decrease in the negative value, that is to say it's less negative, and yet that still doesn't reach the threshold. But maybe along comes a slightly larger stimulus, and now all of a sudden the millivolt value passes the threshold, and something very dramatic happens here. We have a depolarization, and we get a sharp rise through the 0 point of neutrality up until positive 40 or thereabouts. This is our action potential occurring, and then after we reach about positive 40, we again begin to repolarize. We actually overshoot our initial value, and then we return once again to our resting state. Now that might seem fairly complicated. What you might find fascinating is that all of this happens within an interval of just a few milliseconds. Here we're showing it across 5 milliseconds. There are some neurons that have been tracked to be as fast as a 1 millisecond interval. That is, this uptick and the down portion of it and back to resting state can all happen as quickly as 1 millisecond. This particular neuron is displaying a 5 millisecond interval for its action potential. An important idea about the action potential is that action potentials obey the all or none rule. That is to say that if the stimulus exceeds threshold, the nerve or muscle fiber will give a complete response. Otherwise, there is no response. As you can see over here, we mentioned earlier that there was some response from this particular neuron, but it didn't reach the threshold. Here's another slight change in the resting potential, but it's not enough to trigger the threshold. If we get up to threshold, we get this all or none response. Some researchers like to invoke different metaphors here or different analogies, and one that can be interesting is the analogy of a toilet flush. So, just like a neuron has an all or none response with respect to its action potential, the same thing is true with a toilet flush. If you press very lightly on the toilet handle, the toilet will not flush. If you press just a little bit more firmly, still nothing, but if you hit some kind of a threshold, you get the full flush. Now, interestingly, the toilet itself does not flush any more vigorously if you push the handle harder. It just goes through its normal routine, regardless of how much magnitude you have on your push of the toilet handle. Another interesting component about the toilet flush analogy is that it has its own refractory period. That is, immediately after you flush the toilet, if you try to flush it a second time, nothing will happen. You need the system to reset itself. Well, that's also captured here. If you had another stimulus coming in right after this first one, that second stimulus might not be registered because this action potential hasn't completed its cycle, it hasn't yet reset itself, it hasn't had its refractory period. As we'll see, there's one other component to the toilet flush analogy, and that speaks to the unidirectional flow of the water in the toilet and also in a neuron when we have the action potentials moving just in one direction down the axon. So let's take a look at that. We've seen this diagram before, and here we have a lot of detail about the cell nucleus that we're not going to need to get into. But in this red area, we have the soma, or the body of the cell, and here we have the axon hillock. Once the axon hillock releases an action potential, the action potential moves in only one direction. Just like the flush of the toilet water, it's going only downward, and we go down and down and down until we get to the axon terminal. So it's unidirectional, it's all or none, and there's a refractory period. So there are actually several points of contact with this analogy of the toilet flush. Okay, let's now see if we can understand at an even more fine-grained level than the cellular level. We'll begin to look at the molecular level, the level of molecules and also atoms. There are a couple of important principles that are diagrammed here. The first of these is the idea that we have a cell membrane, as we had seen before. This time the cell membrane is vertically oriented. An important principle here is that we have a semipermeable membrane, and that means that certain kinds of atoms or molecules are allowed to pass relatively freely from one side to the next. This is the extracellular space over here on the left. This is the intracellular space. This is the membrane separating the two. So often it is the case, in fact, really for all mammals, we have a very particular kind of semipermeability, and that is that potassium ions, shown here with this K, are able to pass relatively freely inside and outside of the cell membrane. They can go to either side relatively easily. By contrast, these sodium ions and these chloride ions don't have the same kind of passage between the two sides separated by this membrane. So we have a semi-permeability associated with this membrane, and we have the potassium ions basically going through much more easily than the other ones would. That's the first principle. We have another principle that might be very familiar to you, and that is that opposites attract. Say it with me, opposites attract. Okay, there it is. And so you notice that we have some negatively charged ions here, and we have some positively charged ions on this side. We also have some anions. That's what this A is standing for. The anion is negatively charged. Here we're showing a fairly large anion that has 4 negatives, and some of the 4 negatives are offset by these 4 positives, as you see here. Here's 1, 2, 3, 4 positives that offsetting the 4 negatives in the anion. But we also have some anions that have 2 positives linking up to offset 2 of their 4 negatives. But here's a couple of other negatives that don't have an offset. And so we have opposites attracting. You might notice that there's some sodium on this side that's positively charged that is lining up very tightly with the negatives on the other side because opposites attract. Here we have opposites attracting, opposites attracting. They would like to get closer if I can anthropomorphize these atoms, but they can't because we have this membrane in the way, and that membrane is not permeable to chloride nor to sodium. Okay, so we have a couple of things going on. We have a semi-permeable membrane, we have opposites attract, we have one more big idea to enter into this picture, and that is the idea of a concentration gradient. We can define a concentration gradient as the distribution of ions across space. So let's look at this region of space shown in the picture, and you might notice that we have a lot of sodium ions over on this side, and we have a lot of potassium ions over on this side, and we have relatively few sodiums on the right, relatively few potassiums on the left. This is an ion concentration gradient. Okay, the distribution of ions across space is right now very unequal and very helpfully captured in this blue arrow. So we have this sodium, which is positively valenced, and there's a lot on the left side, and as you go from left to right, the number of sodium ions is falling off. And we have the opposite situation here for the potassium ions. Also, note that they're positively charged, and these are going from here right to left. We have a high concentration on the right, a low concentration on the left. So it is true that ions are attracted to their opposite. It is also true that they prefer to run down their concentration gradient. Stated more simply, the ions tend to spread themselves out. So generally speaking, if I can anthropomorphize these potassium ions, they don't like to cluster too many near one another. They like to spread out a little bit and mix it up. But they can't because they've got this semipermeable membrane. One other wrinkle before we move on from this slide is the idea that typically on the inside of the cell, we have a sodium-potassium pump that begins just throwing out positively charged ions. And when it does that, it tends to create a negativity inside the cell relative to the outside of the cell, and that tends to be around minus 70 millivolts, as we had seen in preceding slides. Okay, so to begin to sum up this slide, we have a concentration gradient going on, and we have opposites attracting, and we have a semi-permeable membrane. Also, we have this sodium-potassium pump that is pumping out some positive ions. Generally speaking, it will be pumping out some of the potassiums. Thanks for watching.